A totally different topic would tell us about some genetic traits. And we're going to start with the idea of a pedigree. We may have heard of a pedigree when it comes to dogs or cats or horses, but a pedigree is nothing more than a family tree that shows the relationships of parents and offspring across a couple of generations in order to trace particular genetic traits. This is something that a genetic counselor may actually be involved with, doing pedigrees and explaining inheritance patterns to couples. In this case, the square represents a male, the circle represents a female. Also in this case, the orange coloration represents colorblind, and the half yellow, half orange represents a carrier for colorblindness. Many genetic disorders are inherited in a recessive manner, but we'll talk about a couple of recessive disorders as well as dominant genetic diseases. And in this case, we would call the heterozygote a carrier. Recessively inherited disorders show up in individuals homozygous for the allele. Keep that in mind. Here's a list of the symbols. Usually, whatever trait you're tracing would be colored in the pedigree. So that's why we call a colored in square an affected male, or the colored in circle an affected female. The diamond represents a person whose sex is not known, and the only reason for that may be a miscarriage or spontaneous abortion, something like that, where the individual was not to term and born. We have the marriage line between the male and the female, also called a mating line for obvious purposes. Not all offspring are the product of a marriage. We have two lines going between a square and a circle representing a consanguineous marriage. Consanguineous actually means that the blood is in the family, so that's a related mating line. So that's like cousins or second cousins marrying, which happens and we can denote in a pedigree. The vertical line coming off of the mating line is the offspring line. And then to the right, it shows that identical twins would be a vertical line that splits into two different sections to denote that they were born at the same time. So there's a small pedigree there showing a father, a mother, and four offspring, the male, female, and then there are non-identical twins, the female, and then the male. This also tells us that there are four children, and they are listed in order, meaning that the male to the left is the oldest, the second oldest is the female, and then the non-identical twins are the youngest. Let's talk about a few of these inheritance, a few of these traits and what their inheritance patterns are. Albinism we already briefly discussed with epistasis, but just to more formally define it, albinism is a recessive condition characterized by a lack of pigmentation in skin and hair. If a recessive allele that causes a disease is rare, then the chance of two carriers meeting and mating is fairly low. And in the case of albinism, it is a recessive condition, meaning that the individual will only show albinism if they receive both recessive alleles. Consanguineous matings, which are matings between close relatives, actually increase the chance of such recessive conditions appearing because it may already be in the bloodline or in the family. There are taboos and maybe even laws against marriages between close relatives, but they do happen nevertheless. Cystic fibrosis is another recessive condition, which means only the homozygous recessive individuals show this, and it's a genetic disease. It's actually the most common and lethal genetic disease in the US, striking 1 out of every 2,500 people of European descent. The cystic fibrosis allele results in a defective or absent chloride transport channel in the plasma membrane. This actually results in a thick, sticky mucus that can have the individual having multiple respiratory infections. Individuals with cystic fibrosis don't have a lot of options for treatments at this point. It's like having a really, really bad congestion but it's terminal. It's always present, not only being sick. And they end up getting sick a lot more frequently as well. The point for me bringing up cystic fibrosis is the fact that not only is it the most common lethal genetic disease, but you would not know if you're a carrier because you're not showing any symptoms. I actually have a case where two of my friends were married and they had a child, and their first child had a lot of complications, and they tested her for cystic fibrosis. Although she did not have cystic fibrosis, a genetic test on the parents showed that they were both carriers or heterozygotes for cystic fibrosis. If you do a quick Punnett square, using two heterozygotes, they have a 75% chance of having a child that does not have cystic fibrosis and a 25% chance of having a child with cystic fibrosis. They made the decision to have a second child, but they actually adopted because a 1 in 4 chance was too high for them. Because the child would suffer with cystic fibrosis, they would have a lot of medical bills and a lot of time devoted to physicians, and individuals with cystic fibrosis tend to have a lowered life expectancy in the 20s or 30s. The last of the recessive conditions that I'd like to talk about is sickle cell disease. Again, only homozygous recessive individuals show this disease. It affects 1 out of every 400 African Americans. The disease is caused by a single point mutation, which basically means that there's one single amino acid caused by one letter in the DNA to be different. That results in the red blood cell taking the shape of a sickle, shown at right. The normal red blood cell is spherical. Symptoms in this individual include physical weakness, because sickle cell often leads to sickle cell anemia. Anemia is fatigue and weakness. Also pain, organ damage, and even paralysis. The reason this happens is that hemoglobin, the substance that carries oxygen in our red blood cells, is misshapen. And if a protein is not the proper shape, it cannot have its proper function, because the shape determines the function. So this single amino acid that's different in sickle cell individuals causes a completely different shape of the hemoglobin. The shape of the hemoglobin does not allow it to properly carry oxygen. So that's why we have weakness, because there's a lowered oxygen levels in the individual, and also pain associated with it, because those red blood cells are traveling in the arteries, veins, and capillaries, and they may be getting stuck and actually causing damage to the inside of the blood vessels. So individuals with sickle cell disease have a lot of those symptoms that I just discussed. I wanted to talk about a few dominant genetic diseases as well. Some human disorders are caused by dominant alleles that cause a lethal disease, and they are rare, but they do exist. The first one to talk about is something called achondroplasia. Achondroplasia is a form of dwarfism that's caused by a rare yet dominant allele. Huntington's disease is another dominant genetic disease that is degenerative in nature, which means once this disease has its onset, it's a continual degeneration. Specifically, Huntington's disease is a degenerative nervous system disease, which means the nervous system cells, which are neurons, continue to break down and cannot function. The disease, however, has no obvious phenotypic effects until the individual is about 35 to 40 years of age, possibly older, which explains why such lethal dominant alleles still exist, because by the time of onset, individuals may have already mated and produced offspring and passed those genes on to the next generation. So these exist because they're still being passed on. If you've ever heard of a television show called Little People, Big World, that deals with achondroplasia, and it shows you that there are people that have this form of dwarfism that can have children that are normal sized because they may be heterozygous for this trait. If they have one dominant allele, they will show the dwarfism. However, they can pass on a recessive allele to their offspring and they would not show this genetic disorder. So those are the only two genetic diseases that I wanted to talk about that are dominant. Overall, I wanted to go back to the thought of a genetic counselor. Genetic counselors, once again, provide information to parents regarding family histories for specific diseases. That also means that there are many tests that are able to be done to determine whether or not certain genotypes exist in an individual. Some of these genetic tests can be performed in the womb before the individual is born, and all of them can pretty much be done once they're born. There are tests, for example, that exist that would tell an individual whether or not they're carrying the Huntington's gene. Huntington's, as I mentioned, was a progressive neurodegenerative disease, meaning that you'll continue, once there's an onset, continue to lose control of your muscle coordination. So you would only be concerned and get this test if there's a family history. But that's pretty difficult in order to make that decision if you wanted to get that test done or not, because there's a test that would tell you yes or no, there's a disease that you will get in your lifetime. So genetic counselors are actually there to talk to the individuals or the families before getting a genetic test to make sure they understand not only the inheritance patterns, because we've talked about a lot of inheritance patterns, ranging from the regular Mendelian dominant recessive to all of the non-Mendelian patterns, incomplete dominance, codominance, multiple alleles, pleiotropy, epistasis, polygenic inheritance, and even environmental influences. So the genetic counselor is well aware of this, and their job is to make sure the individual understands these inheritance patterns, especially the ones that deal with disease, and also assess the individual's mental state as to whether or not they're able to receive such news, understand, and process it. For a growing number of diseases, tests are available that not only identify if you have the disease, but also if you're a carrier. And some couples are now making decisions as to whether or not they will have children based on if they're carriers for genetic disease. I just wanted to briefly talk about two tests that could be done while a child is still developing in the womb. The first is called amniocentesis. As pictured at right, amniocentesis is when a long, thin needle is used to remove amniotic fluid from the amniotic sac. So during pregnancy, the baby develops in what's called the amniotic sac. There's amniotic fluid in there, which actually, when the baby is ready to be delivered, the sac breaks and the fluid is released, and that's what they say the water is breaking in a woman that's pregnant. So during development, and this is done usually in the second trimester at about 16 weeks or 4 months of development, the needle would be used to extract some fluid because the fluid would contain fetal cells. Those fetal cells have the DNA of the developing baby. They can do those genetic tests on those cells to see if genetic diseases exist. The DNA from the fetal cells is karyotyped, and you can see a couple of different things, and I'll discuss a karyotype momentarily. So you can do genetic tests using the DNA from the fetal cells, or even do a karyotype. And pictured at the bottom right is a karyotype. Once again, you're ordering the chromosomes from the largest to the smallest to check to see if the chromosome number is correct, as well as the gender of the individual. The second test that can be done prenatally is called chorionic villus sampling. This is done a little bit earlier in the first trimester at about 8 to 10 weeks of development. And this is where a sample of a structure called the chorionic villus is removed and tested. The chorionic villus is actually a structure that develops into the placenta. The reason you're able to take a sample of that and diagnose the developing fetus is because the chorionic villus cells actually contain the same genetic material as the fetus, making them fetal cells. The DNA from those cells would then be used for testing or karyotyping. This is done a lot earlier because in some individuals they may choose to abort the fetus if there are major complications that are discovered. However, we're not here to talk about the ethics of these tests. I'm just letting you know about the tests and why they may be performed. So once again, the chorionic villus is a structure that develops into the placenta, and it has the same genetic material as the developing fetus. It actually comes from the zygote. Once the zygote starts to develop, part of the zygote will replicate into the fetus, and the other part will replicate into the amniotic sac and the developing placenta. So finally, just a reminder and an explanation of karyotypes that's pictured at right. The karyotype is just a picture of chromosomes prepared during mitosis so that they're condensed, and they're arranged from largest to smallest, so 1 to 22, which are the autosomes, and then the 23rd pair are the sex chromosomes. So karyotypes can show you about 3 different things. Number 1, they can show you the number of chromosomes that the individual has. For humans, the key number is 46. We'll talk next lecture about some of the times when 46 is not the key number, but the individual still survives. The gender or sex of the individual can also be shown because you can see in this genotype there are 2 X chromosomes, meaning it's a girl. However, if there was an X and a Y, it would be a male. And then finally, you can see if there are extra or missing pieces of chromosomes. By comparing the chromosomes side by side, you can see if one is longer or shorter and know if something has happened to that individual. Finally, there are other techniques that are tests done on the developing fetus, such as ultrasound, which you may be more familiar with, and fetoscopy, that will show the developing fetus. They assess fetal health while they're still developing inside the uterus. There are other tests that would have, and what's pictured at right, I think is 3D ultrasound, where you're actually able to see features with depth. Remember, not only can tests be done while development in the fetus, but also there are standardized, routinely performed genetic tests that are performed directly after birth. And listed here are 2 of them. One is for phenylketonuria, or PKU, and another is for congenital hypothyroidism. Both of these can be treated, but must be diagnosed early. If they're diagnosed early, they prevent severe conditions such as mental retardation from setting in the individual because diagnosis is made at birth. And there are accommodations that can be made for those individuals to lead long, healthy lives with those genetic disorders. But at birth, the newborns are tested for a plethora or a multitude of genetic diseases. With these review questions, this ends the video lecture portion. You can review these as many times as you need. I do recommend that you are able to do a genetic problem for every type of non-Mendelian inheritance pattern. I will give you a take-home quiz that has a lot of genetics problems ranging the multitude of non-Mendelian genetics that we've discussed. And as you can imagine, I'd like you to have a good handle on these concepts that were discussed in the video lectures. So thank you for allowing me to try the video lectures, and I hope that you found them helpful. I hope that you are able to use them easily, and I hope to do this more in the future if this was beneficial. Thank you, and I'll see you next class.